Good afternoon, ladies and gentlemen, and thank you for a lovely introduction. Today, I'm here to represent the BioVendor Group and its ideology. My presentation will be about the world of microRNAs and their amazing functions and potential they have for science and diagnostic. In the end of my talk, I would like to introduce novel methods that BioVendor has developed for their quantification. MicroRNAs are small and short, non-coding RNA molecules which are encoded by genomic DNA. They were discovered back in 1993 by a group of Victor Ambros and his colleagues. They were actually studying a function of a LIN4 gene, which is important for a larval development of a worm C. elegans. And they were very surprised when they found out that this gene was not coding a protein, but it was coding an specific RNA, which didn't code for the protein. And they found out that this RNA has a regulatory function. And when they searched for the sequence, they found out that these short RNAs are complementary to the LIN14 mRNA, messenger RNA. And by that time, it was known that there is a regulatory mechanism between the LIN4 and the LIN14, but the exact pathway was not described. So they found out that this LIN4 microRNA is binding directly to the messenger RNA of a LIN14, and this is what they name a type of an antisense mechanism. And this antisense mechanism caused that the LIN4 knockout worms were stuck in the baby phase, while the LIN4-14 double knockouts, they were aging too soon. But back to the microRNA. As I mentioned, they are encoded by genomic DNA, and they are transcribed by RNA polymerase into the primary microRNA. These primary microRNA are then subsequently processed. You can see that they have removed the tail, the 3' and 5' ends, and they are turned into the precursor microRNA. This precursor microRNA is shuttled to the cytoplasm, where it is finally changed and made into the mature microRNA. The mature microRNA is actually a single-stranded molecule, which is identified by the RNA-induced splicing complex, otherwise known as RISC. This RISC complex, based on the type of the RNA which is entering, is either psi-RISC, in case that the small interfering RNA are a part of it, or miREIA, in case that it's a microRNA which enters the RISC complex. Once the RNA is included into the complex, it serves as a guiding molecule, and it helps to guide the complex to the target messenger RNA. And then it actually either leads to the translational repression or to the mRNA destabilization. To modulate this process, Cell has actually developed several methods. So it either restores the microRNA function by the double-stranded microRNA mimics, or it inhibits the microRNA function by a single-stranded inhibitor RNA. So now the question is, when you know the basic principles around the microRNA, why to study them? First of all, microRNAs are extremely stable in comparison to the messenger RNA, and they are highly expressed and easily detectable in the biofluids. The expression profile is very sensitive to the various cellular and organism changes, and based on which can actually reflect everything from the tumorigenesis to the inflammation. They have a huge potential of regulation all the major well-known signaling pathways. They are right now becoming a spotlight of biomarkers for diagnosis of various human disease. And their importance is eventually also reflected in the worldwide exponential growth in the number of scientific publications that are involving the analysis of a microRNA profile. MicroRNA are mostly active in the cytoplasm, but inside the cell, they actually can play a role at all the possible places. They can either directly work at the nucleus, where they can bind to the DNA, or they bind to the RNA, which is just being transcribed. They can well shuttle on the microtubule, so it can be a part of endoplasmatic reticulum or the Golgi apparatus. They are crucial for all the basic cellular activities like differentiation, development, or homeostasis. The dysregulation of any of the microRNA functions is associated with various diseases, as I already mentioned. And the interesting thing about the microRNA is that it can be actually released from the cell into the intracellular matrix, where it serves as a chemical messenger and it can mediate cellular communication. So, as I mentioned, it is very important in all the well-known and crucial signaling pathway. So, we all know the p53, which is one of the well-known tumor suppressors. It's working as a transcription factor, and it actually targets its further target. And the microRNA is also very important in this pathway, because it's not only that it can directly affect the p53, but the p53 can be also affected by interaction of a microRNA and the p53 targets, the interaction partners. So this is just an example, and there are many of them. It's even the Wnt pathway, when microRNA is crucial for elementary procedure like epithelial to mesenchymal transition. It's also very important for T cells maturation and differentiation. It's crucial for PD-1 pathway, which is also important for development of immune response. And in this one, it can actually even blocks directly the receptors which are on display at the outside layer of the cell. So this is the scientific part, but as I mentioned, it also has a bright future as a biomarker. So microRNAs are highly expressed and highly stable in the biofluids, and as I mentioned, their expression is very sensitive towards all the possible inflammations or tumorigenesis which are happening inside the body. So it influences the profile. So once we have a panel of specific targets of microRNA, we can always track down their expression, and based on the changes of this expression pattern, we can propose a very early diagnosis. And we can even put it further, because right now, for example, the microRNA might be an interesting target for a COVID situation, because just very recently published paper found out that some of them are crucial for COVID to bind with it and make it possible to enter the cell. So it's even possible to inhibit these microRNAs and use it as a treatment target. So now when we all know why is it important and what is it good for, we really need to face the biggest question, and the ugly truth is that the size matters, ladies and gentlemen. And as I mentioned, the microRNAs are extremely short, and I don't know whether you've ever tried to design PCR primers for something like this short template, but I did try and it never worked. And the reason is that the polymerase really doesn't like to work this way. So what most of the competitors did, they actually artificially prolonged the original RNA sequence. But we know that once you artificially prolong something, it's never that perfectly, and it usually costs you some specificity because you're using universal primers either in the reverse transcription or in the PCR itself. So the biovendor comes with a different approach, and we developed actually two very interesting methods. One is called miREIA, because this one is using a hybridization procedure, and we created a single-stranded DNA, which is complementary to the microRNA which we are screening for, and this DNA has a biotin label. And once they are hybridized together, they create a hybrid, and this microRNA DNA hybrid is recognized by the antibody, and then which is captured to the microplate, and then you just go on with the standard ELISA protocol, and by the reaction of the HRP and the substrate, you can measure the quantity, the concentration of microRNA. But if you prefer to stick with the PCR method, there is a novel method which is called the Two-Tailed RT-PCR, and this one was developed in the cooperation of Biovendor and Tata Center together with some of the excellent researchers here in the Vestec. And they actually designed this special RT primer which has two hemiprobes which are shaped together, so they only recognize specifically the targeted microRNA and only the mature version of the microRNA. And then the procedure is very similar to the standard PCR, and this attitude actually brought us incredible specificity for our product. And this is just an example of a family of microRNAs. And if you look at this arrangement, you see that the majority of them differs in one or two nucleotides. So you need very specific assay to really distinguish between them and not to measure all of them. And if you look at some competitors, really those which are less different, they really have a strong overlap. So you are never really sure what you are measuring. And therefore, I'm glad that I had the opportunity to introduce these novel methods which are really highly sensitive and highly specific. And for the two-tailed PCR, there is even possibility of a multiplexing, which is also quite novel for these methods. And in case that you would be really interested in the microRNA screening of your project, there is also a possibility to use these special panels because they identify either 176 or over 300 microRNAs linked with the pathways, and you can really build up a lovely heat maps where you can see which microRNAs are mostly influenced by your model or your project, and then you can particularly focus by the two-tailed PCR, for example. So I would like to thank you for your attention.